Professor Mahapatra is going to speak about open surface microfluidics in cancer research. So please welcome Professor Mahapatra.
Thank you, Subhanjan, and thank you for inviting here. I'm a bit scared to talk about a little bit on the cancer research in front of the biologists. So I'm a mechanical engineer. We work on mostly on the multi-phase systems and on the surfaces. So today I will talk about one of our work on surfaces and its applications on different types of cancer cells. So before that, there is a small lag.
You can.
Okay. Oh, but it is not sync. So I'm saying something else.
Okay.
Yes. Small delay. So we are from Chennai. IIT Madras is in the southern part of the country, and we have a very beautiful campus. So please visit us whenever you are coming to India. So I worked on, in general, surface engineering, which has applications in creating surfaces of different structures, starting from micro nano structures. We do weighting patterns and use it for droplet microfluidics, droplet manipulations, use it in paper microfluidics for diagnostics, for identifying different analytes, and use it for different systems, for example, detections of adulterants in milk and other liquid products. Also use it for different heat transfer applications. Being a mechanical engineer, we work on different heat transfer problems like condensation, frosting, boiling, evaporescence, and so on. We use it for different devices like development of heat pipes, vapor chamber, thermosiphons, and also as a group we worked on several fundamental problems of particle interacting with liquids, liquid-liquid systems, and so on. Personally, I'm also interested in modeling of systems, for example, identifying the collective dynamics of, let's say, active systems. It can be mixing and segregations of particles. It can be the prey-to-prey dynamics, also the crowd control. So we do crowd control modeling, and also did some experiments on human crowds and trying to understand how human behaves in a crowded situations. We do microswimmers modeling, as well as I collaborate with our colleagues there to understand how bacteria behaves on systems, and then try to simulate and mimic the situations. But today we will talk about the biological systems, where we mainly worked on 3D cell cultures like the spheroids and organoids, which most of you were talking about, and also how we can do drug delivery on single cell level and also do cell patterning. So this is the overview of the talk. I will not talk about all of these in detail. This talk is mostly done by Gayatri, who did PhD with me, and then now a postdoc at University College London, and my collaborator is Tuhin in Engineering Design Department of Mechanical Engineering in IIT Madras. So before starting, I will talk about open surface microfluidics, which is slightly different than the microfluidics we are talking about here. So where we are talking about open surfaces, where we have an interface, of a solid surface and a liquid interface. And then we are talking about a liquid which is sitting on a surface and it gives some contact angles. And based on that weighting, before that I should define what the wettability means. Wettability is the liquid's ability to spread on the surface, and we generally identify the wettability of a surface by defining the contact angle at the triple line of the 3-page contact line on the surface. So this depends on very fundamental aspects, how or what type of weighting it is, what type of surface, whether it's a solid, liquid, or it is a semi-solid or a gel type of surface, and in case of a solid surface, whether there is a roughness presence or not. So depending on that, we will have different contact angles. For our understanding here, I will define the contact angles. If it is less than 90 degrees, it is a hydrophilic surface. If it is more than 90 degrees, it's a hydrophobic surface. And the extremes where the contact angle is close to 0 is superhydrophilic, and if it is more than 150, it's a superhydrophobic. So based on that, we define the different surfaces, and that changes the behavior of the liquid as well as the cells. So we'll talk about that. So this is the Young-Laplace equations, which tells us, and as I said, the roughness are important, and based on that we can see different types of surface structures. Based on the roughness scales, we can see different contact angles. Say one thumb rule is, if your surface is hydrophilic, you make it rough, it will become super hydrophilic. If its surface inherently hydrophobic, make it rough, it will become super hydrophobic, and so on. But why these open surface microfluidics are important? Where in these open surface microfluidics, we don't create actually channels. What we create is the contrast of the angles. So there is no physical boundary, it's all the energy barriers. So we will have open surfaces, there are only energy barriers. I will show you a few examples afterwards, and that will dictate the flow. Because if it is a super hydrophobic, liquid will not go there. It will remain within the hydrophilic or super hydrophilic areas. So you just create the energy barriers within the surface. And this has applications in several fields. As I was saying you in the beginning of this, my presentations, it has applications in energy, heat transfer, physics, chemistry, space science, biomedical, and environmental science, and so on. So we can use it for point of care testing, biosensing, disease diagnostics, and so on. So as I was telling, you can create surfaces where there is a energy barriers, and these you can create in two different ways. One is either you can do surface modifications through chemically by changing the surface energies, or physically by changing the roughness. So you create, as I was telling earlier, you create the difference in the roughness scales, so that you will have one side hydrophobic, other side super hydrophobic, one side hydrophilic, other side super hydrophilic. We can create on any material. It depends on your application. So it can be it can be on paper to a metal. On any material we can create depending on the applications, and then we can do weighting patterns. When we are talking about the weighting patterns, it means locally selective modifications of the surfaces. So we have a recent review which talks about these processes and different applications and so on. So with this background, I will talk about how to use these pattern techniques on cancer cells, or how we can do patterning on the cells. So using these patterns of the energies, we can definitely create structures, and we can isolate liquid drops. So similarly, we can also do cells. So before that, so in traditional cell cultures, we do culture cells on petri dishes. So these are bulk populations of cells. But imagine if we can do cell patterns isolated. Like we can do even in the single cell level, if we can isolate the cells and we can use it for drug screening or other applications. So we'll have a large amount of data. So that's why single cell patterns are important, and we can use it by isolating the cells. So as I was telling, so if we can do this single cell patterning, or even few cells patterning, we will understand the cell heterogeneity even in the same cell lines, whether all the cells are similar or it's different. If we can understand, we can identify distinctive cell states, and also we will get a huge amount of data. So the classical approach of doing this cell patterning are either you can use a physical model like stem cell, even microrobots, traps, or even droplet encapsulations, or in a physiochemical methods like photolithography, microcontact printing, scanning probe lithography, and so on. So what we have tried is in the microcontact printing, where it's something similar to a rubber stamp. You take a stamp where you have a protein which will give you a cytophilic areas, like the way I was talking about a hydrophobic and hydrophilic. Here it is a cytophilic and cytophobic. So you create cytophilic proteins which will adhere the cells, and the back side you fill it with some kind of a cytophobic material, say for example fluoride. So you print the protein that will attack the particular type of cell lines, and then the background you fill it with fluorine. So the way we created this, we created a lithography process where we will have a silicon wafer, we use a photoresist, then we did a PDMS casting, and then we did a lift off, and finally we created the stamp. And then we used a protein, the particular type of protein we want, and then we print it on the surface. And then we backfill with glutamic acid. So this is a kind of lithographic stamp that we have used, and we can change the dimensions or the gap between the pillars, and based on that we can create different types of patterns. So the micro patterns we can obtain based on the structures we want, how many cells we want to print, and based on that we can change the pattern size and the dimensions and so on. So before that, say this is a protein stamp, we can create on the, this is with a fluorescence, so we can do the protein stamp and then we can pattern the cells. Say for example, here if you want to do a cervical cancer cells like a Sinha or a fibroblast, you can use a fibronectin to attract it, and or say for MG63 you can use a collagen, and then we can pattern it. Say for example, and this, as these are created using patterns, so we can even print IIT Madras. So you can do any type of structures you want. And depending on the size, you can see here, like with a few cells to almost a single cell level. So we can pattern it, pattern the cells, and any type of cells, depending on if we have a proper protein that we know that will attract, we can print it. So now we know that this is possible to print the cells. We can go up to a single cell level, but what next? Why we'll do that? So what we are trying to do here is, can we do a biomolecular delivery into the cells? So this biomolecule can be siRNA, mRNA, to like enzymes. So my colleague Tuhin, he is an expert in this drug delivery into the cells. So what we are trying to see is this intercellular delivery. Generally, either you can use a diffusion-based process or through a microrobotic kind of direct drug delivery. Rather, we have chosen a process which is known as optoporation. So optoporation is a process where you can create a cavitating bubble by shining a laser into the cells, and that will create a small hole, and you can deliver the drug or whatever the biomolecules you want to deliver into the cell lines. So these are very selective, and you can control it based on the lasers. So for that we have created a titanium microdisc device. So this titanium microdisc device actually helps us to create these nanobubbles or the microbubbles depending on the size and the shape, and that helps us to deliver it. So I'll not go to the details of how to create it. So rather I will see what are the different molecules we have delivered. So for the on the Sinha cells we have delivers like the PI dextrain, even the siRNA enzymes and so on. So which are of different sizes. Say for example, when we have started with a PI which is 668 Dalton, so we are able to deliver it on the Sinha and MG63 with a 5-second pulse range. And similarly, we are able to do dextron and siRNA and enzyme. So which are different in size, and particularly these are very big biomolecules which are able to deliver quite efficiently within the cells. So in a single cell level it is possible, or in a few cells it is possible, but is it possible to do in a co-culture platform? Particularly these are like a microcontact printing, right? So microcontact printing are so far done on one type of cells, not on multiple cells, multiple different types of cells, which are in general very difficult because you cannot stamp two different proteins together. So the conventional technique for this co-culture is either you can use a direct co-cultures, mix the two types of cells and then culture it, or use a Prince1 systems like this, or you can use a droplet encapsulations or like a compartmentalizations. So what we thought is we cannot replicate with a appropriate cellular organizations if we do that. So if we can do a multi-protein patterns of these cellular organizations. But directly multi-protein patterns is difficult. As I was telling earlier, you cannot print two with a stamp and then two different types and then you can do a microcontact printing. So what we did is like a open microfluidics. We will close it and then we will remove the top part. So we used a one stamp which will create a hole here. So we will stamp this and then this will create a microchannels, what we are in general closed microchannels, and we flow and the second protein through it. So one we will print like a microcontact printing I showed earlier, and within these holes we flow the other one. So this will give us, so first one ink you can directly print here, and the second ink will flow through that like the red color here. So the ink 2 will flow through it and it will create a multi-proteins. So as you can see here, these are with two different colors. So one is red and green. You can pattern two types of proteins. So what we did is we have basically created a microfluidic systems. We closed it, then we opened it afterwards. So, but the challenges are how you can do it without any leak and so on. So that we optimized. So basically we created this protein pattern on PDMS. Then we backfilled again with a plurionic. So we have again a cytophilic, cytophobic areas. Now we have two proteins which will attract two different types of cell lines. Now the challenges is what type of cell will adhere to what. So that we have optimized based on the times. So we will see that which type of cells will adhere first. That we will do. That will adhere, and then we will do the second cells and so on. So the say for example, if we want to do NIS3D3 and HG2, so you use a laminin. This 50 microgram per ml is what the optimum values are. So HG2 will adhere first, and then you use fibronactin for NIS3D3. So like that you can patterns, and you can see that we can do multiple or parallel printing of the cell lines. Like we can do a co-culture, we can create a co-culture platforms of different cell lines. So we can do NIS3D3, HG2 with MG63. We can also do with HeLa NIS3D3. Like it can be any combinations possible depending on we have to do with the optimizers, how much time you have to seed the cell, and what combinations of what concentrations of the proteins you have to give. So then we have studied the linear expansion profile. We also did a kind of wound healing, like a scratch assays we have tested, and we have created this for or cultured it till 96 hours to see how it is growing in these co-culture platforms. So now if we have this micro-engineered co-culture platform that can be used for several studies, for example, this human microenvironment mechanism studies, or the disease infection studies, or even a personalized drug screenings, tissue engineering, and so on. So now you can see how this same drug, whether it is working on different types of cell lines or it is very specific to one type of cell. So you can do a lot of studies there. These are all ongoing works. So this is a platform which you can use it for different types of studies. So till now these are all on a 2D structures. These are on 2D surfaces. So now, okay, before that, there are certain challenges on these microcontact printings. So you can always print from a hydrophobic surface to a hydrophilic surface, not the other way. If your surface is hydrophobic, you cannot print it. you cannot print a protein. Protein will not adhere there. So you need a hydrophilic material to print. So there are limitations of these microprinters. So for example, this is a very nice work where you can see that where the contact angle is 0 degree, it will print very efficiently. But if it is like more than 90 degree with the hydrophobic surfaces, the printing efficiencies are very poor. The main challenge is single link per stamp. You cannot use multiple link unless you use the flow type of systems I was telling earlier. And this protein transfer proceeds with a low energy surfaces like the hydrophobic surfaces to always a high energy surface like a hydrophilic or super hydrophilic type of surface. So we have to pre-treat the surface, only then you can do this efficient transfer of the proteins, otherwise it will not move. And there are high prone to errors like the manual errors. So for example, as if you use full manual system, then there is a chance of skipping. As you can see, it's not print exactly the place you want. It will skip a little bit, like our rubber stamp. If you don't press it properly, it will not print. So you need a system. You need a proper mechanisms to do that. So these are some challenges. It is not as simple as I was telling earlier, and there are durability issues because you are talking about proteins. So proteins are not durable as such. So now I think I have some time. So now, yeah, I will go back, and now I will very quickly I will talk about 3D cell cultures because these are all 2D so far we talked about. Now can we do culture on droplets? So that will give us a 3D environment. Say for 2D cell cultures, so far these are robust, these are simple, low cost and so on. But there are certain limitations. So it is restricted to interactions between the cellular and extracellular environment. These are lack of tissue architectures. You don't have anything because these are all 2D planar and so on. And these are inability to represent the complex cellular heterogeneity. So we need a 3D cell cultures. So now if you see experimental convenience, we have the 2D cell cultures, we can create spheroids, then organoids, and finally we can go to animal models and human models. So that is how these studies should be. So what we started with a 2D cell cultures, and then we moved into spheroids, and then organoids and so on. So today I will talk about 3D cell cultures platforms. So among the scaffold-free environment presents, so either we can use the static suspensions or we can use some microwell-based 3D cell cultures platform, or even a magnetic systems which requires complicated setups. So what we thought of creating a simplified lab-based systems which are low cost and which uses a hanging rod. And you can use these different types of droplets. Either it can be hydrogels or it can be just the medium. But if it is a medium, then you have to take care of the evaporation effect. Otherwise, it will evaporate. So you have to take care of that. So I will not go into the magnetic printing. These are already available, commercially available 3D cell cultures technologies by calling like microplates. You can use it for cell cultures. So what we have used is what I started discussing earlier is the weighting patterns. So if we use a weighting patterns, like the way liquid attracts or hit the surfaces, cell also do that. So if you can use a super hydrophobic surfaces, cell will not flow. So this is what I was talking about. Like liquid will not go into the super hydrophobic areas. It will remain confined within the hydrophilic state. Say for example, there liquid is spreading, but this is only because of the energy barriers. There is no physical barriers. So these microfluidics, when we are talking about open surface microfluidics, you don't have any physical barriers. These are all energy barriers. So here, like the way we can pattern liquids, we can pattern cells. So what we did is, we created a very simplified method. So this is a carbon, and that you create by just lit a candle. And you don't burn it properly. If you do like if your burning is improper, then it will create a lot of soot. So that these soots are biocompatible because these are carbons. So what you have to handle is this should not come out from the surface. So to do that, you create for any material, say for example here we are using a glass slide, you can use other material, and then you give a PDMS layer on top of it. You can use some other polymers, and then don't cure it properly, hit the carbon soot, and then with that burning, with the temperature itself, it will cure. These particles will embed within the PDMS and it will cure there, and that you can use as a super hydrophobic surface because carbon is hydrophobic, and that will give you a roughness, so it will become super hydrophobic. And PDMS is hydrophobic in nature. So as I was telling earlier, you take a surface which is hydrophobic, make it rough, it will become super hydrophobic. So you will have a super hydrophobic surface, you will get a contact angle around 160 degrees. And these are very stable. Very low cost, you don't need any clean room, you can do it at your lab. And then what we did here is use a CO2 laser and your applet. Whatever the design you want, whether it's a circle, square, or whatever design you want, you applet the surface. The limitations is the size. So with the CO2 lasers, you can go up to 50 micron. And for a droplet, that is good enough. It depends on what spheroids you want, what size of the spheroids you want. But these are very controlled. You can create 50 to 500 micron quite easily. Just create a pattern on any softwares like a Corel or anything you do, and then you print it. Then we can use a de-wetting, like instead of using properly pointing the liquid in each of these, you use the de-wetting techniques where you put a drop and then you slide over these areas. So your each area will have a particular amount of liquid to hold. So it will hold that. Where you can calculate, like from the force balance, you can calculate how much volume of the liquid it can hold, and that will give you the size of the liquid droplet that can stay there. Now, if you want a 2D cell cultures, you use it as a sessile drop. But if you want a 3D cell cultures, you do it upside down. So then it will be a hanging drop. So if you want a 2D cell culture platform, you do it like this as a sessile drop. It will adhere to the surface. You can do a 2D cell cultures like the way you are doing it on the Petri dish. But if you want a 3D cell cultures, you can do it with the hydrogel also. This is a platform, you can use it hydrogel, then you shine a UV light to cure it a little bit. Or you can use a 3D spheroid platform. You put the droplets, put it upside down, and you can see the difference in the spheroid formations. So if you don't use this wetting confined areas, your Sinha or any 3D3, it will not form the spheroids. It will form more like a stick. You are biologist, so you can understand it better, but that is what we observe. But if you use a droplet, if you use a hanging drop, these are more circular. You can say for example, the Sinha spheroids, your sphericity index is close to 1 always. So it is almost a perfect sphere. Whereas if you don't pattern it, you will see the size is different. It is not close to 1, these sizes are more like a flat shape rather than a sphere. So we have used it for Sinha as well as in a 3D3, it is always a spheroids. And then we done this confocal and SEM for these spheroids. Okay. So now from all of these, the main conclusions, we can use this cell patterning for manipulating and study intercellular processes. We can use it for large scale staining, for drug candidates, genetic modifications, and other therapeutic interventions. We can use multi-protein patterning for co-culture models, creating of complex tissues and organ models by mimicking interaction between different cell types. We can use this low-cost wetting patterns for multimodal cell cultures. Like here I'm showing only the results of one type of cells. We can do cell-cell interactions, spheroid-spheroid interactions by creating two spheroids and then merging it with the droplets, which are quite easy to do. We are working on that. And these are these can be used for advancement in cancer research, drug discovery, and regenerative medicines, and so on. So But there are several roadblocks, particularly the minimizing the expert interventions and ease of use is the grassroots level and the users. And there are many of these are all in the academic laboratories. These are not coming out as a technology and should be validated and emerge as a commercially viable product. I think only few of these are coming into the market and others can use it. So there are several roadblocks which we have to overcome. Only then we'll be able to use it for actual drug screening and drug applications, regenerative medicines and so on. And for that, we need definitely an interdisciplinary team. Like this type of workshops are important, where people from different backgrounds can come together and work together for this type of research. So I want to acknowledge my collaborators, my students, my funding agencies, and with that, thank you. Any question? Yes.
So very interesting talk. I'm Mahapatra from Madras Institute of Liver and Living Sciences in Delhi. So my question is that you have used the FG2 and the fibroblast cell line. If you use any kind of immune cells, have you tried? Because with the patterning with the immune cell would be difficult,
Yes,
especially in the cancer research. Nowadays the macrophages are coming up. And the mono and the max are coming up. Those are the true, really the cancer kind of therapeutics. So can we use your patterns along with the immune cells, like the hepatocyte tank along with the
True.
immune cells?
Pattern, you are talking about a
Patterning of the immune cells, instead of doing only the two cell lines, you get the one hepatocyte or the any cell line, you get the immune cells.
Yeah,
And
so
then we see the interaction between.
yes, we can. So we can do that. I have not shown all the results here, but yes, you can. So it all depends on what type of proteins you are using. Say, we will talk about it afterwards. So yes, yeah. Yeah, sure. We'll discuss this. So it depends on your seeding time, because this is the adhesion process, right? So if you use different types of cells, so you have to use a proper proteins, and that should adhere to that, and then you can use them.
And how long we can do this?
So if you are talking about these type of platforms, these are only one-time use. You can use it only once. You culture it, then you cannot repeat it. But if you use a waiting patterns, like the way I was talking about, you can use it multiple times.
No, for how long? Means for 7 days, 10 days?
Yeah, you can use it. 7 days is fine. We have used up to 7 days, yeah. That you can use.
Thank you. Any other questions? Yeah.
Just one short question. It's very interesting and appealing and open, these open surfaces microfluidic. But one drawback, for instance, when you're using droplets, is the high ratio of surface over volume.
Yes.
And the huge evaporation rate.
Yeah, so that is what I told earlier. Like if you can control that, so there is a way to control. So you have to create an environment where your evaporation rate will be very minimum. So you have to saturate the medium. Saturate with, let's say you have a medium where your hanging drop is. If you're using hydrogel, there is no issues of evaporation. But if you're using liquids, so then you have to saturate the environment with that medium, so that your evaporation rate is very low. Otherwise, we have used another type of approach where you can add the medium approach. So you need a porous material at the top, and you can give a drop of liquid from the top. If you know, and what is the
agriculture medium, and then it should merge
with another medium you can use.
But you can use oil. For
Yes,
instance.
yes,
You can go
exactly,
with
exactly.
oil, right?
You can use that.
You can use oil.
So you need a droplet in a liquid medium.
Yeah.
You can do
Are
that.
you going to use an environmental chamber or something? Like
No, even in an environmental chamber it will evaporate. So that is what I was telling earlier. You have to saturate the medium with that. Even in an environmental chamber, you cannot saturate with 100% liquid of that medium, right? So you have to create it separate chambers. So we have tried that, So even with the oil we were doing, I'm not showing all the results here.
Well, I think there are some papers where they are using
Yes,
this,
yes,
they
yes.
cover it with oils, but they are using traps.
Yeah, so
traps, and
here,
they drop
here
in the
these
drops
traps
and
are,
then-
exactly, so here these traps are waiting patterns.
Yeah.
You don't have to create a physical traps.
Yeah,
These are
that's
all
what-
energy
that's
traps,
very,
yeah.
very interesting. And also,
These are very low cost, you can use any material to do this.
and to use the, I mean, your technique to make the PDMS super hydrophobic by using the soot particles is very, very nice. But you can also use like sterilization to make it super
So,
hydrophobic.
yes, but if you use silenizations, those are toxic for cells.
Oh.
For water drops, yes, you can do this, but you cannot use it for cells. These are not biocompatible. So silents are not biocompatible. And also there are PFAS and so on, so you cannot use it. Only possibilities, you can use silicon dioxide nanoparticles and then you coat it with some kind of other polymers, with some copolymers you can use. But that is also, first of all, those are not durable, but soot is very durable. You can use it for many times. We have used it for 10 times. You culture cells, you wash it again, then do it, it works.
What is the size of typical size of nanoparticles made of soot?
So these are in few nanometers.
Few
So,
nanometers?
yeah.
Okay, that's fine.
Yeah.
Okay, thank you very much.
Yeah, very nice presentation. So, what I can understand from this is that we have the traps and you have the cells. Now, is there a way to culture the cells back? Means say, or extract the cells and get RNA and protein in abundant amount from your the open stripes that you have shown, or is it like that? These are we can see the association and the movements and different other aspects, but not at the molecular level as a downstream assay.
Well, I don't know whether it's possible or not. Probably you guys can know better than me. But if you can do it on single cell or any other cells which you are culturing on a petri dish, if that is possible, here also it should be. Well, that is what my answer. But I
May I
don't know.
respond to that?
Yeah,
You are
sure.
talking about the stripes, right?
Stripes.
So if the cells you are using which has a differential trypsinization timing,
Yes.
then you can actually do it separately.
Because if you can do it on a co-culture platform, otherwise here also you can. That's what my understanding. But
Okay, and I feel personally that this might be also, can we think of looking into, I think somewhere you have mentioned about metastasis, like looking into interaction between two completely different types of cells.
Yes,
I understand,
yes, yes.
I understand we need to optimize the adhesion
Yes.
aspects of the cells,
Correct.
but we can probably look into a transendothelial between
Yes,
the
exactly.
endothelial cells migration, and those might be a very nice platform because it's an open one, and hence it might be a very nice platform. It's nice. Maybe we can talk later.
Sure.
Thank you.
yeah. Okay.
Okay, so maybe it's time to go for the.
Thank you.
Okay, for the speakers of the session.